Can poor sleep hurt muscle growth and testosterone? If you train regularly, you probably already know the basic formula for building and maintaining muscle. You train hard enough to give your muscle a reason to adapt. You eat enough protein to provide the building blocks they need to repair, and then you repeat that process consistently over time. But there is another part of that equation that is often treated as optional: sleep. Most of us understand that bad night's sleep can make us feel tired the following day. Our training might feel harder. We might have less energy. We may not perform quite as well. But what if poor sleep is doing something deeper than simply making exercise feel more difficult? What if inadequate sleep changes what is happening inside your muscles after you've finished training? Researchers have found that sleep deprivation can affect muscle protein synthesis, alter hormones involved in recovery, and potentially create an environment that makes maintaining and building muscles more difficult. And some of these changes have been observed after surprisingly short periods of sleep restriction. In this video, we are going to look at what happens to your muscles when you don't get enough sleep. We'll examine how sleep affects muscle protein synthesis, testosterone and cortisol, whether exercise can protect against some of the effects of sleep restriction, and what scientific evidence tells us about sleep, recovery and muscle growth. Because training doesn't end when you leave the gym. Your body still must recover from the work you've done, and a significant part of that recovery happens while you're asleep. I'm Journal of Figures, and I'd like to thank you for taking time to watch this video. If you enjoy the content, find it useful, or think it may help somebody else, consider hitting the like button, subscribing to the channel, and sharing it with someone who may benefit from it. One of the clearest ways to understand why sleep matters for muscle recovery is to look directly at what happens inside the muscle tissue when sleep is taken away. And that is exactly what researchers led by Séverine Lemon investigated in a study published in Physiological Reports in 2021. The researchers wanted to answer a relatively simple question. Can just one night without sleep change the rate at which the human body builds new muscle protein? Before looking at what they did, we need to understand something called muscle protein synthesis. Your muscles are constantly undergoing a process of breakdown and rebuilding. Old or damaged proteins are broken down, while new proteins are created to repair and maintain muscle tissue. The process of creating those new muscle proteins is called muscle protein synthesis. This becomes particularly important when we exercise. Resistance training provides a stimulus to the muscles, while adequate nutrition provides the building materials. Muscle protein synthesis is part of the biological process through which the body repairs and adapts to that stimulus. The researchers recruited 13 healthy young adults, 7 men and 6 women. Each participant completed 2 different conditions using what is called a randomized crossover design. A crossover study means the same participants experience both experimental conditions. Instead of comparing one group of people who sleep with another completely different group of people who stay awake, each participant effectively acts as their own comparison. This can be useful because differences between individuals, such as genetics, body composition, and metabolism, are less likely to distort the comparison. In one condition, the participants were allowed a normal night of sleep. In the other condition, they underwent 1 night of total sleep deprivation. That distinction is important. This wasn't a study of people getting 6 hours instead of 8 hours. The participants in the sleep deprivation condition were kept awake for the entire night. We should be careful about applying these findings directly to somebody who simply loses an hour or 2 of sleep. During the following day, the researchers measured several things. They examined participants' hormonal profiles, including testosterone and cortisol. They also took muscle samples and measured what is known as the fractional synthesis rate of muscle protein. Fractional synthesis rate is essentially a way of estimating how quickly new proteins are being produced within the muscle over a particular period. And the results were significant. Following the night of total sleep deprivation, muscle protein synthesis was approximately 13% lower than during normal sleep conditions. Testosterone concentrations were approximately 24% lower following sleep deprivation, while cortisol concentrations were approximately 21% higher. Why does that matter? Testosterone has numerous functions within the body, and it is involved in maintaining muscle tissue and supporting anabolic processes. Anabolic simply means processes involved in building longer molecules and tissues. Cortisol also has essential functions. It helps regulate metabolism, blood pressure, immune activity, and the body's response to stress. Cortisol itself is not a bad hormone. We need it. But the combination observed in this experiment, lower muscle protein synthesis, lower testosterone, and higher cortisol following sleep deprivation, suggested that the body's internal environment has shifted away from conditions normally associated with building and maintaining muscle. The researchers described this as an increase in what they called a pro-catabolic environment. Catabolism refers to a biological process that breaks larger molecules down into smaller ones. When researchers describe an environment as pro-catabolic, they are not saying that somebody suddenly starts losing visible amounts of muscle overnight. They are describing physiological conditions that may favour breakdown relative to building. Interestingly, the researchers did not find a significant difference in the gene markers of muscle protein degradation that they measured. That is an important detail because it prevents us from exaggerating the findings. The study did not demonstrate that one sleepless night caused participants to lose 18% of their muscle. It did not demonstrate that their muscles suddenly began breaking down dramatically. And it certainly did not demonstrate that one bad night's sleep permanently damages your ability to build muscle. What it showed was a much more specific. After one night of complete sleep deprivation, these 13 healthy young adults had an approximately 18% lower rate of muscle protein synthesis during the measured period, alongside lower testosterone and higher cortisol. The researchers concluded that a single night of total sleep deprivation was sufficient to produce what they describe as anabolic resistance and procatabolic hormonal environment. But there are important limitations. Firstly, this was a very small study involving only 13 people. Secondly, the participants were healthy young adults, so we cannot automatically assume that exactly the same response would occur in older adults, people with chronic health conditions, or every other population. Thirdly, this experiment involved total sleep deprivation, which is considerably more extreme than the partial sleep restriction most people experience in everyday life. And perhaps most importantly, this was an acute experiment. It tells us what happens following one night without sleep. It does not tell us exactly what happens to muscle mass after months or years of consistently sleeping 5 or 6 hours per night. But that is precisely why the study is useful. It provides a potential biological mechanism. If inadequate sleep repeatedly suppresses processes involved in muscle protein synthesis or altering the hormonal environment surrounding muscle recovery, then the researchers have a plausible pathway through which chronic sleep loss could eventually influence muscle maintenance, recovery, and body composition. One sleepless night isn't going to erase months of training, but the experiment demonstrates something important. The recovery process doesn't begin when you walk back into the gym. It is already happening. or potentially being impaired while you sleep. The first study showed us what can happen to muscle protein synthesis after a single night without sleep. But total sleep deprivation is an extreme situation. Most people who are chronically sleep deprived are not staying awake for an entire night. They are simply going to bed too late, getting up too early, and repeatedly sleeping fewer hours than their body needs. The question becomes much more relevant to everyday life. What happens to muscle protein synthesis when people still sleep every night but simply do not get enough? A study published in the Journal of Physiology in 2020 by Nicholas Sainer and colleagues investigated exactly that question. The researchers wanted to determine whether several consecutive nights of restricted sleep could reduce the body's ability to synthesize new muscle proteins. They also wanted to know whether exercise could potentially protect against some of those effects. Before looking at the experiment, we need to define something called myofibrillar protein synthesis. Muscle contains several different types of proteins. Myofibrillar proteins are the proteins that make up much of the contractile machinery within our muscles. These include proteins such as actin and myosin, which interact with one another to produce muscular contraction. Myofibrillar protein synthesis, often shortened to MyoPS, is therefore the process through which the body produces new myofibrillar proteins. This process contributes to repairing, maintaining, and remodeling skeletal muscle. Over time, the balance between muscle protein synthesis and muscle protein breakdown helps determine whether the muscle tissue is maintained, gained, or lost. The researchers recruited 24 healthy young men. Before the main experiment began, everyone completed 2 nights of controlled baseline sleep. That allowed the researchers to establish more consistent sleep conditions before introducing the experimental intervention. The participants were then divided into 3 groups. The first group was the normal sleep group. These participants were given 8 hours in bed each night. The second group was the sleep-restricted group. They were given only 4 hours in bed each night. The third group was also restricted to 4 hours in bed each night, but these participants additionally completed 3 sessions of high-intensity interval exercise, as stated in the study, but I will keep referring to it as training going forward, commonly abbreviated to HIIT. High-intensity interval training involves repeated periods of relatively intense exercise separated by periods of recovery. Rather than exercising continuously at the same intensity, participants alternate between harder efforts and recovery periods. The researchers matched the groups for several characteristics including age, body mass index, habitual sleep duration, and VO2 max. VO2 max refers to the highest rate at which somebody can take in and use oxygen during intense exercise. It provides researchers with an indication of a person's aerobic fitness. So matching participants for VO2 max helped reduce the possibility that major differences in fitness between groups would distort the results. The experimental period lasted 5 nights. That means that sleep-restricted participants weren't completely deprived of sleep. They were still given 4 hours in bed every night, making this experiment different from the Le Mans study we just discussed. The researchers also took muscle biopsies before and after the intervention. A muscle biopsy involves removing every small sample of muscle tissue so that researchers can examine what is happening within the muscle itself. And the results showed a clear difference between the groups. The rate of myofibrillar protein synthesis was significantly lower in the sleep-restricted group than in both the normal sleep group and the sleep restriction plus exercise group. Put simply, 5 nights with only 4 hours in bed reduced the rate at which these participants were producing new myofibrillar muscle proteins. But there was another particularly interesting finding. The participants who experienced the same sleep restriction but performed 3 sessions of high-intensity interval training maintained myofibrillar protein synthesis levels comparable to the normal sleep group. That suggests that exercise provided a sufficiently strong anabolic stimulus to counteract the reduction in myofibrillar protein synthesis observed during sleep restriction in this experiment. The word anabolic simply refers to biological processes involved in building larger molecules and tissues. When we describe exercise as providing an anabolic stimulus, we mean that it activates processes that encourage the body to repair, remodel, and potentially build muscle tissue. But we need to be careful with how we interpret this finding. It does not mean that exercise cancels out the consequences of inadequate sleep. This experiment investigated one specific outcome, myofibrillar protein synthesis, over 5 nights. It did not demonstrate that exercise protects against every physiological consequence of sleep restriction. Poor sleep can affect many other systems, including cognitive performance, hormonal regulation, immune function, glucose metabolism, and recovery. The message is not sleep 4 hours and compensate by exercising harder. The finding is much narrower and more interesting than that. Under the conditions of this study, high-intensity interval exercise appeared to preserve myofibrillar protein synthesis despite substantial sleep restriction. The researchers also investigated several molecular signaling pathways involved in muscle protein synthesis and degradation. One of those was mTOR. mTOR is a signaling protein that plays an important role in regulating cell growth and muscle protein synthesis. You can think of it as part of the cellular signaling network that tells muscle tissue when conditions are appropriate for building new proteins. They also examined AKT, another signaling protein involved in pathways controlling metabolism, cell survival, and protein synthesis. Interestingly, despite the lower myofibrillar protein synthesis observed in the sleep-restricted group, the researchers did not find corresponding changes in the AKT and mTOR markers they measured. They also examined markers associated with protein degradation and autophagy, and found no significant changes between the groups. Autophagy literally means self-eating, but biologically it refers to a cellular recycling system. Cells use autophagy to remove and recycle damaged or unnecessary cellular components. This creates an important distinction when interpreting the study. The problem observed in this experiment appeared to be reduced building rather than clear evidence of accelerated breakdown. And when we compare this study with the Lemon study, an interesting pattern begins to emerge. Lemon and colleagues found that 1 night of complete sleep deprivation reduced muscle protein synthesis by approximately 18%. Sanner and colleagues then demonstrated that 5 consecutive nights with only 4 hours in bed also reduced myofibrillar protein synthesis compared with normal sleep. These were different experiments using different methodologies, so we should not directly compare the percentages as though they were measuring the same thing. But both studies point in the same general direction. Whether sleep was removed completely for 1 night or substantially restricted across several nights, the body's production of new muscle proteins was reduced. There are, however, important limitations. This study involved only 24 participants, and every participant was a healthy young man. We therefore cannot automatically assume the same results would occur in women, older adults, highly trained athletes, or people with chronic medical conditions. The sleep restriction was also severe. 4 hours in bed per night is considerably less than the amount of sleep recommended for most adults. Someone regularly sleeping 6.5 hours rather than 8 hours may not experience the same response. The intervention also lasted only 5 nights. This study cannot tell us precisely what happened to muscle mass after months or years of inadequate sleep, nor does a reduction in protein synthesis over 5 days automatically mean that someone will visibly lose muscle. What it does provide is another piece of mechanistic evidence. Building and maintaining muscle requires more than simply lifting weights and eating protein. Your body must convert those nutritional building blocks into new muscle proteins. And this experiment suggests that when sleep is severely restricted, that process itself may become less efficient. If you are training hard, consuming sufficient protein, and wondering whether sleep really matters for your results, the study provides a useful answer. Training provides the stimulus. Nutrition provides the raw materials, but adequate sleep appears to be part of the biological environment that allows your muscles to use them effectively. When we bring the evidence together, the message is clear. If you care about building muscle, maintaining muscle, or recovering properly from training, sleep is not something that sits outside the equation. It is part of the equation. The first study we looked at by Lemon and colleagues demonstrated just how quickly sleep loss can affect the environment inside the body. After only one night of total sleep deprivation, muscle protein synthesis was reduced by 18%, cortisol increased by 21%, while testosterone decreased by 24%. That does not mean one sleepless night suddenly causes you to lose your muscle, but it does demonstrate that the biological processes involved in muscle repair and recovery can change remarkably quickly when sleep is removed. The second study by Sanner and colleagues took that question further. Instead of one night without sleep, participants experienced 5 consecutive nights with only 4 hours in bed. The researchers found that myofibrillar protein synthesis, the production of proteins responsible for the contractile structure of our muscles, was significantly lower in the sleep-restricted group than in those receiving 8 hours in bed. But there was another fascinating finding. The participants who were sleep-restricted but also performed 3 sessions of high-intensity interval training maintained their myofibrillar protein synthesis at levels comparable to the normal sleep group. That does not mean you can simply exercise your way out of chronic sleep deprivation. The study was small, involved only 24 healthy young men, lasted just 5 nights, and measured a specific aspect of muscle metabolism. Exercise maintaining muscle protein synthesis does not tell us that every other consequence of inadequate sleep disappears. What it does show is something much more useful. Sleep and exercise do not operate independently. Training provides the stimulus for adaptation, nutrition provides the raw materials, and sleep helps create the biological environment in which recovery and adaptation can take place. If you're spending hours in the gym, progressively overloading your muscles and carefully thinking about how much protein you're eating, but consistently sacrificing sleep, you may be neglecting one of the variables that helps turn all that effort into results. You don't need perfect sleep every night. Life doesn't work that way. There will be late nights, early mornings, and periods when getting enough sleep simply isn't possible. The evidence we've looked at does not suggest that an occasional bad night destroys your progress. The concern is when inadequate sleep becomes your normal routine, because muscle isn't built while you're lifting weights. Training creates a stimulus. What happens afterwards determines how effectively your body adapts to that stimulus. Train properly, eat properly, recover properly, and sleep properly. You get one body. Fuel it properly.